Good afternoon. My name is Paul South. I'm a chemist in the Office of Food Safety and FDA's Center for Food Safety and Applied Nutrition. The title of my presentation today is FDA's Regulatory Program for Chemical Contaminants in Food. Today I will briefly discuss the statutory authority and enforcement options used by FDA to address chemical contaminants in food. I will also provide a description of FDA's monitoring programs that address chemical contaminants, including FDA's Total Diet Study. In addition, I will discuss several of the chemical contaminants that FDA monitors in food, including recent work involving arsenic, and finally describe some of FDA's international efforts on chemical contaminants. The Federal Food, Drug, and Cosmetic Act provides FDA with regulatory authority with regard to adulterated foods that are in interstate commerce or delivered for introduction into interstate commerce. Specifically, Section 402 of the Act defines when a food is adulterated. Under Section 402A1, a food would be considered adulterated if the food bears or contains any poisonous or deleterious substance, and if the substance is added, FDA must show that the quantity of the substance in the food may render the food injurious to health. Or, if the substance is not added, FDA must show that the quantity of the substance in the food ordinarily renders it injurious to health. Therefore, if a substance is deemed added, then FDA need only show that it may render the food injurious to health in order to regulate consumption of the food containing the substance. The may render standard means that there is a reasonable possibility of injury to the consumer. However, if a substance is considered not added, FDA must go further and show that the substance would ordinarily render the food injurious to health before it can regulate its consumption. Under Section 402A1, FDA has different enforcement options to address chemical contaminants in food that affect public health. FDA's approach to addressing chemical contaminants with no FDA established levels is to handle instances of contamination on a case-by-case basis. For example, for chemical contaminants found in food, FDA conducts a health hazard evaluation by reviewing relevant information, including levels of the contaminant in the food, the type of food, consumption of the food, and all relevant toxicological information for the contaminant to determine the level of a contaminant that would represent a potential health risk such that the food would be deemed adulterated and subject to enforcement actions under the Federal Food, Drug, and Cosmetic Act. Another enforcement option available to FDA, but rarely used, is establishment of tolerances. This option has been used only once for addressing PCBs in food and feed. One reason this option is rarely used by FDA is because it involves rulemaking, which is long and often time-consuming for both establishing, revising, and revoking tolerances. FDA's monitoring of food for chemical contaminants includes several different programs, including FDA's Total Diet Study, FDA's Toxic Elements and Radionuclides in Food Compliance Program, FDA's Pesticide and Industrial Chemicals Compliance Program, and FDA's Mycotoxin Compliance Program. The Total Diet Study, or TDS, sometimes called the Market Basket Study, is an ongoing FDA program that determines levels of various contaminants and nutrients in foods. From this information, dietary intakes of those analytes by the U.S. population can be estimated. Since its inception in 1961 as a program to monitor for radioactive contamination of foods due to fallout from nuclear weapons testing, the TDS has grown to encompass additional analytes. A unique aspect of the TDS is that foods are prepared as they would be consumed, referred to as table-ready prior to analysis, so the analytical results provide the basis for realistic estimates of the dietary intake of these analytes. The study designed for FDA's TDS involves the collection, preparation, and analysis of 280 foods and beverages. Using levels found in TDS table-ready foods and consumption amounts obtained from national dietary surveys, FDA calculates dietary intakes for chemical contaminants and nutrients for the total U.S. population, as well as for 14 age and gender population groups. Each sample is tested for approximately 400 analytes. Analytes measured under TDS includes over 400 pesticide, 43 industrial chemicals, 13 radionuclides, and 18 elements. Chemical contaminants that have been added to the program include dioxin in 1999, acrylamide in 2003, and perchlorate in 2006. For TDS, there are 4 regional market baskets collected each year. Each market basket is made up of 280 foods collected in 3 cities in each region. The 3 samples that are collected in each region are composited for each individual TDS food, then analyzed. TDS intake estimates are calculated by multiplying analyte concentrations in each food by the amount of food consumed. Intake estimates are calculated for 14 age gender groups and for the total U.S. population. TDS intake estimates are used for different purposes. For example, they are used to provide estimates of background dietary exposure, to monitor the impact of regulatory actions, to identify potential health hazards, to assess national nutrition monitoring efforts, and to provide support for international development of food standards and risk assessments. One toxic element that FDA monitors in food is arsenic. Inorganic forms of arsenic, including the tri- and pentavalent forms, are the arsenic species of greatest toxicological concern. In contrast, organic forms of arsenic, which is the predominant form present in seafood, demonstrates little toxicological potential. Recent concerns have been raised regarding the occurrence of inorganic arsenic in certain foods, including certain juices and rice products. Arsenic is an element that occurs in the environment from natural causes, but may also be added from human activities, such as smelting and mining. Arsenic can be found in air, water, soil. 2 forms of arsenic are found in food. The organic forms are generally non-toxic, often associated with seafood. The inorganic form is more toxic. Arsenic exposure has been associated in humans with cancers of skin, urinary tract, and lung, as well as skin lesions, developmental effects, neurotoxicity, and diabetes. Arsenic has been found to occur in a wide variety of foods, including rice, vegetables, fruit juices, including apple juice. Concern about arsenic in apple juice in particular was raised by consumer groups and the media in 2011. FDA had previously completed a hazard evaluation for arsenic in apple juice for internal use in 2008 that established a level of concern of 23 parts per billion inorganic arsenic in apple juice. Starting in 2011, FDA initiated new work related to arsenic in apple juice, including a survey of retail apple juice samples. 94 single-strength apple juice samples were collected. 4 samples were found to contain total arsenic above 10 parts per billion. No samples contain inorganic arsenic above 10 parts per billion. Other work included a new draft quantitative risk assessment for arsenic in apple juice. The purpose of the draft quantitative risk assessment was to assess risk from consumption of lower levels of inorganic arsenic, typically seen in apple juice, and to inform development of an action level for arsenic in apple juice. Endpoints were different than those previously used and included urinary tract and lung cancer due to chronic and lifetime exposure. In July 2013, FDA released a draft quantitative risk assessment and a draft guidance for industry including a draft action level. The guidance identifies an action level of 10 parts per billion inorganic arsenic and states that FDA intends to consider the action level in addition to other factors when considering whether to bring enforcement action in a particular case. There has also been increased interest in arsenic in rice. Rice is high in arsenic relative to most foods. Specifically, inorganic arsenic comprises from 25 to 90% of total arsenic in rice, depending on factors such as rice type and growing area. Advances in speciation technology over the last several years has allowed measurement of inorganic arsenic in rice. In addition to the quantitative risk assessment and guidance document, in 2012 and 2013, FDA released new data on arsenic levels in rice, including approximately 1,300 results for different types of rice, as well as rice products, including infant and toddler cereals, pasta, grain-based bars, snacks, desserts, and beverages, including beer and rice wine. This table shows results for the analysis of rice. To highlight a few points, you can see that the mean inorganic arsenic level in brown rice is higher than white rice, and that the basmati white rice tends to be lower in inorganic arsenic than the non-basmati long grain white rice. Note that the full rice data set is available on the FDA website. FDA is currently working on a draft quantitative risk assessment for rice and rice products. This document will be peer-reviewed and released for public comment soon. The risk assessment will help inform risk management and policy options for arsenic and rice. Future possible follow-up activities at FDA include additional sampling of selected rice products, as well as fruit juices, seafood, other grains, mushrooms, and baby foods. Other research will include cooking and preparation studies to see if changing the way that rice is washed and cooked can lower levels of inorganic arsenic, as well as what effect these changes have on rice's nutritional status. FDA also participates in the Codex Alimentarius, which is an international organization under WHO and FAO that establishes international standards and codes of practice for food. FDA participates in the Codex Committee on Contaminants in Food. This slide shows the results for arsenic of this year's meeting of the CCCF and the CAC, which endorses decisions of the subject committees. The committee approved maximum level for inorganic arsenic in polished rice of 0.2 milligrams per kilogram that was adopted by the Codex Commission. Ongoing work includes development of a draft maximum level for husked rice ranging from 0.25 milligrams per kilogram to 0.4 milligrams per kilogram, though no decision was reached on this issue. The electronic work group will reconsider this issue for next year. Finally, the committee agreed to move forward with work on a code of practice for the prevention and reduction of arsenic contamination in rice. Another chemical contaminant that FDA monitors is acrylamide. Acrylamide is a chemical contaminant that is formed in certain foods during processing. Specific processes associated with acrylamide formation include frying, baking, or broiling. Acrylamide can be found in a variety of foods, including potato chips, French fries, bread, cereal, sweet potatoes, asparagus, cocoa, and coffee. Levels of acrylamide in food can vary in production lots and between lots, between different products, and between different food types. Acrylamide is a genotoxic carcinogen, meaning it is a cancer-causing agent that can damage DNA. FDA has measured levels of acrylamide in different foods and has posted data on acrylamide levels in approximately 2,600 food samples. The following table shows some of the top 20 foods by mean acrylamide intake. The intake levels are a function of both the acrylamide level found in the food as well as the amount of the food consumed. FDA has also been involved in research to study the process by which acrylamide is formed in food. For example, FDA has studied the effect of cooking temperature on acrylamide formation in French fries. FDA studies indicate that in French fries, acrylamide formation increases with higher frying time and temperature. In addition, acrylamide formation is highly correlated with darker French fry color. Similarly, in toasted bread, light toasting causes very low acrylamide formation, while dark toasting causes low to moderate acrylamide formation. In addition, scraping toast to remove brown surfaces has been found to reduce acrylamide levels. Another chemical contaminant that FDA monitors in food is dioxin. Dioxin and chemically related compounds, referred to collectively as dioxin-like compounds, or DLCs, can be found in food-producing animals. Because DLCs tend to accumulate in fat, consumption of animal-derived foods, including meat, poultry, eggs, fish, and dairy products, account for approximately 95% of human exposure. Studies suggest that dioxin-like compound exposure may lead to a variety of adverse health effects, including reproductive and developmental problems, cardiovascular disease, increased diabetes, and increased cancer. Reduction of emissions from numerous combustion processes have resulted in significant decline in reported dioxin emissions in the United States. EPA estimates emission reductions to be more than 90% since 1987. The following graph provides exposure estimates for dioxin in TDS foods for the total U.S. population and for 14 age and gender population groups. In 2012, the Environmental Protection Agency released a dioxin hazard assessment addressing the non-cancer health effects associated with dioxin exposure. Included in the EPA dioxin hazard assessment is a reference dose. FDA has posted a dioxin questions and answers document on the FDA website that provides information on dioxin and the food supply. Another chemical contaminant that is monitored by FDA is lead. There is little to no margin of safety for lead, and therefore some level of risk associated with even low levels of exposure. Lead is of particular concern for the developing fetus because the placenta presents a minimal barrier to the movement of lead from maternal to fetal circulation. In addition, in infants and younger children, food consumption per body mass is greater as compared to adults. Another concern with lead is that children absorb lead more readily than adults, by a factor of about 4. Moreover, the immature status of developing organs, including the brain, kidneys, and liver, make the fetus, infants, and children more sensitive to the toxic effects of lead. FDA's Total Diet Study has been analyzing food for lead for decades, and Total Diet Study results, as shown in this slide, indicate a large reduction in dietary lead intake. A number of factors, including FDA's ban on the use of lead-soldered cans for food, are thought responsible for this large decrease in dietary lead intakes. In 1994, the California Department of Public Health identified imported Mexican candy contaminated with lead that had migrated from ink used on candy packaging. In addition to the package, FDA also discovered that Mexican candy made with certain ingredients, for example chili and tamarind pulp, contained the highest lead levels than that found in domestic candy. To address this issue, in 1995, FDA issued its first guidance level for lead in candy of 0.5 parts per million. In 2006, FDA issued a revised guidance level of 0.1 part per million for lead in candy likely to be consumed frequently by small children. Another chemical contaminant that FDA monitors in food is perchlorate. Perchlorate is both naturally occurring and man-made. For example, it is found naturally in certain fertilizers, but it is also manufactured for use as a propellant in rockets and missiles. Perchlorate has been detected in numerous surface and groundwaters in the U.S. used for irrigating crops. Perchlorate has been found in crops grown using water containing perchlorate. One issue associated with perchlorate is that it can inhibit iodide uptake by the thyroid. Impairment of the thyroid can affect the fetus and newborn, resulting in delayed development and decreased learning capacity. FDA has been testing TDS and non-TDS foods for perchlorate since 2006. The following table provides exposure estimates generated from total diet study foods for different age and gender population groups. FDA also analyzes food for mycotoxins. Mycotoxins are low molecular weight secondary metabolites produced by certain fungi during the growing, processing, transport, and storage of certain food. Mycotoxins occur naturally and are difficult to avoid. Various toxicological outcomes occur from consuming mycotoxins. Many mycotoxins are stable to heat and food processing procedures. FDA monitoring activities consist of targeted sampling of foods that are susceptible to mycotoxin contamination. Through this monitoring program, FDA has derived estimates on the incidence, levels, and dietary exposure that are used for risk assessments and for establishing guidelines. To address mycotoxin in food, FDA has established limits, including action levels and guidance levels, for different mycotoxins in food. For example, for all products except milk, FDA has established an action level of 20 parts per billion for a mycotoxin called aflatoxin. For milk, FDA has established an action level of 0.5 parts per billion for a specific aflatoxin called aflatoxin M1. FDA has also established an action level of 50 parts per billion for a mycotoxin called patulin in apple juice and apple juice products. FDA has also established a guidance level for a mycotoxin called deoxynivalenol in finished wheat products of 1 part per million. FDA has also established a guidance level for a mycotoxin called fumonisin in different corn products that range from 2 to 4 parts per million. In addition to these monitoring efforts, FDA also participates in international committees to address chemical contaminants in food. As mentioned earlier, FDA participates in the Codex Alimentarius Committee on Contaminants in Foods, a committee established under the Codex Alimentarius. The Codex Committee on Contaminants in Food establishes maximum levels for contaminants and naturally occurring toxicants in food and feed, elaborates codes of practice for prevention and reduction of contaminants and naturally occurring contaminants, and risk assessments by the joint FAO and WHO Expert Committee on Food Additives. While not officially part of the Codex structure, the joint FAO and WHO Expert Committee on Food Additives provides independent scientific expert advice to the Commission and in its specialist committees like the Codex Committee on Contaminants in Food. The most recent meeting of the Codex Committee on Contaminants in Food was held in the Netherlands this past April. Issues addressed included setting maximum levels for lead in infant formula, inorganic arsenic in polished rice, and fumonisins in maize and maize products. With that, I'd like to conclude and thank you for your attention. Note that information on FDA's regulatory program for chemical contaminants in food is found on FDA's website. Or if you have specific questions, you can contact me. My email address is listed. Again, thank you, and I'd be happy to answer any questions.
Well, thank you very much, Paul, for that very informative presentation on chemical contaminants. We have time for just one or two questions. And the first question is, was the level of arsenic found in baby food of concern to FDA?
FDA is looking at different foods, including baby food. We will be posting data like we have in the past for rice and rice products. At this time, that information is being reviewed by the scientists, and it will be posted. I don't have that information at this time.
There is another question that says, how is left-centered data handled by FDA? So when setting action levels of maximum levels, how is left-centered data, left-censored data assessed for analytical test results?
Less sensitive data?
Less censored,
Oh, sensory data.
Yeah, censored. Left-censored data, which I don't understand the term.
Well, sensory data, when doing risk assessment and action levels, what we look at not only is the contaminant that we find in the food, the levels that we're looking at, but we will also look at the type of food that the contaminant is being found in, as well as who consumes that food, sensitive populations that consume that food, as well as any other issues that involve the toxicological properties of that chemical. But in regard to sensory,
Censored, C-E-N-S-O-R-E-D.
censored data, well, we don't really censor data, so yeah, I thought that was sensory.
We'll move on to another question, and this is our last question, is what action is taken if FDA finds that a food exceeds the levels, the acceptable levels?
Well, again, it depends on if it's a domestic or imported food. We can seize the food. There could be an injunction. The food could just simply be detained at the border if it's an imported food and sent back to the original country.
Well, again, I want to thank you, Paul, for this most informative presentation, and thank you for participating in the webinar, and thank you for the questions that came from the audience. Oh, wait a minute.